Hi, everyone. My name is Tyler Morgan, and I'm going to be talking today about investigating neural responses using fast non-selective MRI and EEG. This is part of the session for non-bold fMRI, and I will add in the no disclosures soon. So just to start off, there's a long history of trying to understand whether MRI can record neuronal signals directly. Hemodynamic fMRI has proven extremely useful for non-invasive measurements of human brain activity, but the potential of replacing indirect blood-based measurements with direct neuronal measurements promises to increase a lot of the issues that we kind of associate with fMRI. And it would give us increased temporal resolution as well as better interpretive power, etc. And like I said, this has kind of been a long history of trying to understand whether these problems are solvable. And these include attempts to try to capture neural activity in things like neural current imaging or NC-MRI, magnetic source imaging, cell swelling tracking, as well as more recently, an attempt to look at neural spiking directly using Diana. And I'm just going to briefly introduce it, although I think a lot of people in this room are already familiar with it. In 2022, there was a publication of an effect that was termed Diana fMRI, and it outlined a potential sensitivity to neural activity directly, just like what we were talking about previously. And what was also really impressive about it was that it utilizes a pretty simple GRE sequence that really the only major change was to swap the phase encoding and repetition loops using what's called a 2D scan line method. Then this made it so that they could sample extremely quickly throughout time courses of neural activity at 200 Hertz in that study. But a single image required as many trials as it does phase encoding lines in order to create those images. And so if you wanted repetitions, you basically had to multiply that by the number of phase encoding lines that you have. And this is very prohibitive for things like human studies. So in our work today, we aimed to record Diana signals in human subjects with non-selective sequences. So in other words, we're not actually recording images, but we're only recording time courses from each MR receiver coil. In other words, to put it another way, we're not using any kind of encoding gradients. We're only recording the pure NMR signal. This does not require trial repetitions for phase encoding lines, and it means that we can kind of take full advantage of trying to get as many trials in to surpass what is normally prohibitive in these types of studies, which is the SNR, the temporal SNR of these. So in this proof of concept study, we aim to investigate whether fMRI signals carry neuronal information, but importantly, that's not our end goal. This is basically aiming to establish whether these signals exist at all and leaving it to follow-up work in order to spatially resolve those signals. And to start off, we basically did a series of experiments where we just played very similar types of experimental stimuli that they did in the TOW study or TOY study, meaning that we just used event-related design. So auditory beeps and light flashes were used here. And basically what we find is that in Diana signals, we can see fluctuations directly after the playing out of some kind of experimental stimuli. We also simultaneously recorded EEG to try to understand whether those signals matched up. As you can see here on the left-hand side for auditory tones, the tones don't look necessarily the same as the EEG studies, but they are similar in some ways. And interestingly, between the two types of stimuli that we use, auditory tones at 1,000 Hertz and 2,000 Hertz, the responses looked distinctly different. And that wasn't necessarily the case for the auditory stimulation paradigm that we were using. At the same time, we also did a series of experiments where we would play out visual beeps or visual flashes. This is essentially just a VEP style recording within the fMRI scanner. And so basically what you can see here is that there is very distinctly a response right after the light is flashed into the person's eyes. But what you'll also notice is that if you look at the peak times, this is essentially associated with a light flash. And basically what this is doing, or what we believe that this is doing, is causing a blink artifact that's really, really difficult to separate from the functional response. And in order to kind of dig a little bit deeper and understand whether we can kind of circumvent this issue of having people blinking during the responses, I know this is a really beautiful slide. This is essentially an explanation here of the frequency tracking experiment, but just verbally for the time being, what this involves is a series of block designs where we flash checkerboards for people at different frequencies to try to understand whether the responses look distinctly different in each of those cases. So in other words, can these actually track the frequencies that we're looking at? And so we used 5 different stimuli, 4 of which I'm going to show today. This is basically flashing a checkerboard at 1 Hertz, 6 Hertz, 10 Hertz, and 15 Hertz. And what we find when we look at the spectrogram response functions that we recorded are that there are responses that are associated with these things. And importantly, as you move through the different frequencies, the responses actually shift in their frequency responses. So here you can see down at the bottom, there's a little bit of response that's associated with the 1 Hertz stimulus. And this is a little bit of an interesting case. The analysis method that we're using, a multi-taper method, might be cutting off a little bit of the 1 Hertz frequency response. But when we go to the 6 Hertz flashing checkerboard, we can see that there is a distinct response during the entirety of the block to the fundamental frequency in that frequency. To the fundamental frequency of the checkerboard flash. As we move up to 10 Hertz and 15 Hertz, we can see shifts in the kind of the hot spots of the frequency responses. And although we're kind of losing power in the responses at those frequencies, there still seems to be some tracking to those frequencies, both in their fundamental and in their first harmonic responses. And this has been previously reported in MEG and EEG results, but never in fMRI. So this is kind of faster than we believe BOLD can ever accomplish. And just as kind of a secondary portion of this, we ran all of these experiments on a phantom as well, just to understand whether this was something to do with the type of experimental setup that we were using, and we didn't find any responses. Mind you, this is a lot less data, a lot fewer runs. This is 10 runs, so just about 2 hours of these. And we believe that if we continue pushing that, we can get them into the kind of same response ranges. And at the same time, I'll also be showing some EEG results. These are stuff that Pete and I are working on today, so those will be added in hopefully in the next few hours. But we're hoping that we can kind of understand the relationship between those EEG frequency responses and the fMRI responses that we're looking at. So here we have shown preliminary evidence for event-related and frequency tracking signals in human fMRI. These experiments utilized a non-selective set of sequences with fast TR and multiple echoes because they're basically FID experiments. And the next steps that are involved in this type of project are to further explore the ability for sequences to actually resolve human neural activity and the development of spatially resolved sequences that can actually tell us the localization of these types of signals. And with that, I will thank everyone.
You're at 9 minutes and 56 seconds.
Okay, that's that's good. Yeah, because it'll go smoother when I'm not explaining what I will be explaining.
So Tyler,
Mhm.
if I could ask a question. So I like your phantom results. I think that it would be, so why is it, so I mean, it looks like the phantom results are like, is that the same scale of power change? I mean, it's sort of like, I mean, it seems like this data obviously is more noisy because it's less averaging, but should that necessarily be that? I mean, it looks like that's there's a lot going on there that's noisy, but in the other ones, there's much less.
Yeah, it's a very good point. So my experience so far, and I'm by no means an expert in frequency analyses. Um, as we added more and more subjects to our human data set, the overall changes that we were seeing initially when we were only at like 4 or 5 subjects was in the range of like hundreds of percent difference in power change. And then as you added more and more subjects, this started to kind of squeeze down into a much, much smaller range. And I think that if we match the same number of runs for the phantom experiments, we'll probably see it squeeze down just as much. I suspect that these types of fluctuations are probably not dramatically different than these ones.
Right.
It's just a matter of that's something that I really have to do because I don't think it really shows the point that I'm saying
Yep.
until that's the case. So yeah.
Yeah, if there's some way to display that in terms of statistical significance,
Yeah.
then it would disappear.
Yeah, I definitely, I mean, one of the things that I'm doing this week will be to just to match those. And I think that at that point, if we're still seeing really noisy results, then something else is going on and we can discuss it a little bit. But I suspect that that's the easiest way to deal with that rather than having a statistical explanation.
Yep. Yep. Okay.
Kind of the question here. So like for your 6 Hz stimulation, the spectrum looks like you not only have, for example, response at 6 Hz, 12 Hz, but you also have a bunch of other high frequency response, right?
Yeah, that's very true. I don't really understand why we're getting, I mean, I know that even in MEG and EEG, you tend to have frequency responses all the way up the spectrum from whatever your fundamental or first harmonic frequencies are. I don't understand why that is. Pete, maybe you can add a little bit of detail to that.
A lot of it's just what we're filtering out. If you show the Meg paper, if you just have it handy, you can see that we see exactly this pattern in the MEG results.
So why?
Not
Why do you see
but, yeah.
Why do you see the same results in MEG? I mean, like what's actually going on there? Is it some smearing of harmonics, or is it
I don't think anyone's ever really taken a stab at explaining away why we see it in sort of the imaging that's not cortical implanted electrodes. It's probably different areas coming together, smearing across the different sensors.
Yep.
But yeah, to my knowledge, no one's really taken a stab at saying like, okay, why do we get it going all the way up the spectrum?
Yeah.
Are people able to see the?
Yes.
Okay. Yeah. So that's the same explanation that I have is I don't totally understand why the frequency response has so much upward drift, but it seems to.
Yeah, I think it would be worth, just having a pop-up of those images in your talk to just say like this kind of mirrors what others have seen in larger scale studies as well with the same stimuli or nearly the same stimuli.
Okay, yeah, that's
yeah.
My hope is that the EEG results will be that, but
yes,
but
we'll
yes,
Yeah, my thought is also that maybe to prime the audience, because they maybe haven't looked at this sort of data. maybe even show like that MEG plot, like say, hey, this is what they see with MEG. And just,
Sure.
5 seconds, like, and then they're sort of primed to look to know, like, okay, that's what MEG looks like. And then I think it would make it more impactful. Yeah.
Okay, that's helpful because, yeah, I mean, what we're doing is a direct replication of that plot, so I might as well show it.
Yep.
Fernando?
Hey, Tyler, thanks. I just maybe a little bit of like a suggestion. There is a lot of information in this spectra. And there's the issue of, like in the lower frequencies, they seem to be like more centered in the stimulation frequencies, but like the 15 hertz one is like, to me, like very surprising. So maybe if you're going to show like the MEG results, you could give some hint or make some point about why this distribution of power doesn't seem to match at all the stimulation frequencies specifically at that range. Because it's sort of puzzling, so maybe there's good reasons for it. Probably there are. Maybe you also see that in your MEG. Maybe it's even there and I didn't see it because it was a bit fast. But that point, that connection might be useful because that's something that popped up like to me.
Okay, thank you. I just so I'm on the same page, you're saying, here, I'll pull it back up. You're asking why do the frequencies not match exactly, or you're asking which part?
So essentially, for example, if you look at the 15 hertz,
Uh-huh.
it matches like very poorly. It's all the harmonic 2, the main frequency, the stimulation frequency.
You mean
And-
it's like not right under the line?
Yeah, it does. For example, on top, it does. It's not
Okay.
even. It's not even in the range of the blob. You know.
Sure, that's
So that's sort of surprising because you expect sort of a concentration of energy sort of in the stimulation frequency and its harmonics, but that does not seem to be like you see that. But you see something more like it in other frequencies. So I was wondering maybe some point can be made about what's going on there.
yeah, absolutely. This is another thing that Pete and I are kind of wanting to get done. So all of our experiments in the frequency tracking one required actually using a computer. And this means that the frequencies that CyclePy is giving us may not be exactly the frequencies that we're telling it to give us. And so one of the things that we want to do is just confirm that that's actually the case by essentially just running it with a photodiode in order to know the actual frequency. At that point, yeah, if it's the case, then yeah, we wouldn't have a very good explanation for why it's slightly off. But if that's the actual frequency, then that might tell us more. So yeah.
This is also more of a presentation comment, but a few slides earlier when you have the time series. Yeah. So like for an EEG audience, they might be reading this better, but especially since this is more of an MRI audience, I kind of see, in both the MRI and the EEG, I see two signals that are pretty much opposite each other, that there's something going on and kind of everything averages to 0. So I think I'm not sure if it needed to change visualization, but I think it's just a little bit more explanation about what people are looking at here. Because I think if someone's not as used to looking at EEG, the amount of signal and what's meaningful or not is harder to understand.
Yeah, that makes sense. Yeah, I'll definitely spend a little bit more time on trying to explain it better. What's the part that you're saying averages out to 0?
I mean, like even like the EEG half, like you have the purples that go down, the greens that go up, and I just see two signals that are like, is it just flipped? Is it like, these are things that like I kind of know from the EEG literature, but this isn't an EEG audience. And then for the MRI, like I see a bunch of stuff near 0 and, a certain number of trials above, but there's no mean, there's no significance, there's no error bars. So it's hard to say
Oh,
what
okay,
you're
yeah.
referring to here.
Then I do need to explain it a little bit better. So each of the colored lines is it's not a trial, it's a spatial location. It's,
Okay.
yeah, so the maps above correspond to what
Okay.
each of those lines is at that given point.
Okay.
But
yeah,
I'll explain that better.
Yeah, definitely explain it better.
There's also a tiny little head in the upper left corner, like near where it says 100 or 200 on the left, that shows where those coils are in the MR and where the EEG channels are.
Okay, so
just-
maybe even pull that out of the subfield and just make like a giant thing on the left and the right of each of those, and then you'll be able to point to it more clearly.
Yeah, that's totally fine.
So
Yeah, I
Yeah.
quickly got sucked into looking at those types of plots.
So
Yeah.
this is more of a style thing, but so yeah, I know in the title you even say Diana. Do you say Diana in your title?
Let's see. No.
So I mean, and then later on you call it the Diana signal, which, you know, my feeling is to not link this to Diana. Just because, you know, with Diana, they were looking at, you know, it was very, very high resolution, and it was sort of corresponding to EEG or implanted electrode signals. And this is sort of like, or, you know, yeah, or rapid or, you know, presumably neuronal. I just think if we should take Diana out wherever we can, because then people be like, what, this is proving Diana's right? No, not really. We're just showing our thing, which is very different in many ways than that experiment. I mean, of
So
course
would
that's
you-
Diana because that's Diana. So, but I think that like when you show like when you do the power spectra and the other ones and our preliminary results, you mentioned that this is
Yeah,
the
I'll
Diana
remove
signal.
it entirely. Do you
Okay.
think I should remove this as an introductory slide as well?
No. This
Okay.
frames it because everyone, this frames it.
Because
But
it was a
that's it.
huge motivation, obviously.
Yeah.
So
Yeah, this
Okay.
is good. This is good to refer to this, but then to say, you know, we don't even know if this signal is the same thing as what they claimed is the signal. So we don't want to kind of like say it is or isn't. I mean, we're looking at our thing and we see something rapid, and that's it. So,
Okay.
yeah.
Sounds good.
And also one last thing, once you get back from the meeting, you better be ready to write this up quickly.
Sounds good.
Okay.
Parash?
Hey, Tyler, this is Boris. Nice listening to you. I have two questions. One of them is related to visual perception and the illusions. Like we have a limit, right? So like sampling of seeing things like 10 hertz, I think, like when like real illusion kind of thing. So we have an upper limit of perceiving things in our visual system. So when you do flickering stimulus like this, I wonder, you are pushing the limits and then it is trying to brain is getting into a different state. So this is kind of visual perception domain that's, I don't want to step too much, but I was curious about that. Second question is related to baselining. It will be helpful if you tell other people, and I think like what type of baseline you applied, and from the EEG literature, there are multiple baselining techniques. One, percentage power change may not be great sometimes. Like there's also log ratio of like pre-stimulus versus post. You take the log post divided by log A kind of thing. So like there are some discussions in the EEG lab discussion list in the past that I thought they opt those type of baselining, and it would be great if you tell people how you baselined here. Thanks.
Sure. Yeah. So your first question was about visual perception and asking I think you were saying that at 15 hertz, you're getting towards the range of lack of actual perception of flickering. I think that that's
Yeah,
how I
for instance,
interpreted it.
like in nature environment, like bees have higher intermittent frequency ranges. So when they fly faster in the air, in the sky, they can see things
Yeah.
better. So they don't blur things. In humans,
Mhm.
I think it was 10 hertz. So when you do 15 hertz checkerboard,
Well,
I am not sure the dynamics may change. Maybe 6 hertz is the best, like detecting those things. I don't know. Like you have to
yeah,
talk to talk to NIMH people about that.
no, I mean, I've definitely spent a long time with visual checkerboards. So one, like, just from like a phenomenological point of view, me doing this task or other people who have done this task, like 15 hertz is not indistinguishable, or it's like 10 hertz and 15 hertz are dramatically different in this experiment, and 15 hertz is hard to look at. It doesn't mean that it's like getting into the range of not being able to perceive the checkerboard. You definitely can. And just to give you like one of the, this is completely anecdotal, but I do think it's a little bit related. At 6 hertz and at 10 hertz, you kind of see continuous frequency tracking throughout the block. In the 15 hertz one, we see this dip in the middle here where it kind of goes away. I suspect it's because it's very hard to look at a 15 hertz checkerboard for 5 seconds.
Yeah, so you know the wheel illusion, like if you see a tractor in the farm fields, you feel like the back tire seems like rotating backwards, even though the car is going forwards, because our
Yeah,
brain is
that's
linking those images. Like you can perceive it, but
for sure.
it reverses in your brain.
Yeah, no, absolutely. And I totally agree that if we included motion in this, then things get very complicated very quickly. Because yeah, you can produce the farm wheel illusion super quick, like really, really strongly with these type of stimuli. But yeah, so because we're completely static, we're kind of immune from a lot of that, unless people start kind of, like you said, hallucinating movement. And that doesn't happen till much later. You typically wouldn't see it at 5 seconds quite yet, but it definitely is a concern. And then your second point about baselining, I totally agree. Percentage power change is a very weird metric. It was used in this particular study because we were trying to match a study that explicitly used it. But I agree that change in dBT, dBdt, or log changes would probably hold some similar information, and probably be a little bit easier to digest. What I worry about, given that it's the fMRI kind of session, is that it should be related at least in some way to measures that people are used to looking at. Like Dan said, we like explain it for people who do fMRI, not for EEG. So,
Yeah, thank you. Thanks.
yeah, of course. Renzo?
Yeah, I have maybe a comment on the dramaturgy. Or like if I imagine, like I recognized it on myself, like I was kind of zoning out at the beginning because you were introducing Diana, which kind of people in the session might know. Then I guess people will sit there and think, is it real or not? Like, are they actually seeing something? The billion-dollar question. And then on slide 6, you show the auditory stimuli, which I think I second that Dan's comment that, like, if you just include one sentence to tell people what they should take home, like that in a given location we have consistent activity across trials or something, then
Sure.
Then this kind of slide is awesome. And then you go to the next time, and then you kind of close the slide, like on the 7th slide, you close it by saying, oh, this is an artifact. So I felt like you're leading me on, kind of, is it real now or not? And so maybe, I know you have a lot of material that didn't make it to the slides, like you looked at the 60 hertz, you looked at Dodi, you looked at the phase cycling and all that. Maybe you can bury this result in one of them, that you say, by the way, we take care of flashing, we take care of DODI, we take care of the trigger delays that we have, and say like, and don't lead me on to say that there's a response that then later I need to kind of revise my kind of frame around it, and then which might, like, if you show all the confounds that you looked at one slide, it wouldn't cost much time, and you still give the impression that you're really in control of the signal and know what you're showing me.
Okay. Yeah, I can. I have these as extra slides currently, but just in case they come in as questions. But I know that in ISMRM, it's relatively difficult to do that anyway. So I'll try to smush them in to just making sure that there's, yeah, that like you said, that we kind of know what we're doing. It's not a very blind thing. We've been looking at it for a while.
So yeah, arguments is that it's motion.
Sorry, Peter, we kind of are losing you a lot.
Okay. Can you hear me now? Hold on.
No worries.
And so that's a problem.
Is he cutting out for everyone else too?
Yes.
Okay. I think this happened last week when he had important things to say.
Yeah, okay.
Now we hear you.
All right,
Yeah,
good.
we can hear you now.
Darn NIH VPN. So yeah, so just basically, yeah, just try to anticipate, as Renzo said, the all the concerns that, you know, it's motion, it's residual bold, it's something weird from the visual stimulus. Yeah, and also of course the you know, you did the phase cycling to reduce any artifacts that way. So yeah, so that's pretty much it. It's pretty straightforward. You can just say it.
Sure.
And
yeah, that sounds good. Yeah, I have had it in my mind like how to respond to this comment that it could be a Hall effect and it's essentially just motion. In my mind, I cannot really, I can't explain the frequency tracking in that way. I can only respond via, I can see that potentially maybe it's the event related effect, but if both are there, then I think that kind of falls apart, but in a, yeah,
so
in an interesting way.
Yeah. So what time is your talk at?
I think it's in the morning, pretty sure. I'm first in the session. I think it's at 9.
What day? What day?
Monday.
So Monday morning. I thought it was non-bold fMRI. That's 1:45 to 3:45.
Ah, yeah, you're right. First thing in the afternoon.
And you're the first
Sorry,
session. Shoot, I might have to miss it because I have to give. So there's a perfusion business meeting, and I usually would never go to that in place of this, but I'm giving a 5-minute tribute to Eric Wong because he's retiring. So, and that's like at the beginning of that perfusion business meeting. So I might,
okay,
that's all right. I'll come to the session afterwards. So
that's good.
All right.
Yeah, I'm the first talk. There is an intro from Camille right before, but I don't know how long that'll last. And I also don't know how big the place is.
Okay.
Tyler, you have seen Farouk's comments in the chat?
Nope.
No, they are nothing major. We can skip it for the time.
What was the refresh? Yeah, so I can comment a little bit on that. And so the frequencies that we chose were 60 hertz is a multiple of all of them. So that's kind of the reason they were chosen. So it's like 4 frames or 3 frames or 6 frames or whatever. Yes, yeah. Humans can recognize extremely fast. It's hard to discriminate above a certain level, but yeah, it becomes difficult to know what's going on at a certain point. Okay, awesome. Any other questions? Otherwise, I'll move on to the second one.
I mean, if anyone's at NIH today and wants to do a straight EEG of stimuli, we would love to borrow your brain. Your hair will
Yeah,
get
totally.
slightly damp.
You can join me. I'm going to do it.
Yoo-hoo, I can have you on the Southwest flight this afternoon.
Okay, I have to admit, I have not practiced this one much yet. I'm not sure if other people know. So Yuhui, this is a power pitch that Yuhui is giving, but he's not able to make it to ISMRM, so I am going to give it for him. And this is a project that we were working on for a while while Yuhui was at NIH. Oops. My biggest problem is always just being able to use a computer. If people don't mind, I'll just do it without the projection mode. Okay, so Renzo, can you keep track of time again?
Yes.
This one's okay. Hi everyone. My name is Tyler Morgan. I'm filling in for Yuhui Chai, and I'm going to be talking today about improving laminar fMRI specificity by reducing macrovascular bias revealed by respiration effects. And neither myself nor Yuhui have any declarations or financial interests that we need to declare. So just to start off, natural respiration variations in fMRI exist. This is an effect that's been long reported, and pretty much all of us experience this who record fMRI data. And here is an example of what we're talking about. Essentially, here's a respiration curve that's lasting throughout a recording run. And if we just look at the times when the breathing is stable versus when there's variation to the trace, we can see pretty distinct changes in the type of image contrast that we're getting. And if we look at the differences between this, you can pretty clearly see that it has structure that is related to the vasculature. And so what we've been doing is examining the spatial correlations between various respiration effects and macrovasculature density distribution. So in other words, as breathing changes, how does its relationship to brain structure also change? So here we have examples of natural breathing, deep breathing, and breath holds. And importantly, there's a correlation between all of the voxels to one another under natural breathing conditions and deep breathing conditions. So in other words, they respond similarly, although they kind of change their values. So correlation between breath holds and deep breathing also is the case. And this can actually be simultaneously related to vascular segmentation. So in other words, what is the explanation of some of these effects and why there are these relationships? And if we look at the vasculature segmentation, we can see that the density is related to the natural breathing variation. So respiration maps from normal fMRI runs closely resemble those to both breath holding and deep breathing. And they show significant spatial correlations with the macrovasculature, which it comes from SWI or QSM images. And what we have been working on is actually using different types of sequences, bold and vapor sequences, in order to understand how the relationships between all of these different types of breathing occur, and how we can kind of remove the effects of some of the largest effects that we see in the vasculature and identify those voxels to remove from layer analyses. So please come to computer number 3 in order to talk about this more. Hope to see you there.
You're at 3.5 minutes, 3 minutes, 21 seconds. And I think you have 2 minutes and they will cut you off after 2.5 or so, I think.
Yeah. Yeah, I'll cut that down a lot. Yihui, do you have comments?
I think you explained it quite clearly, but also maybe too much details in the fourth slide, the second to last one, maybe explained too much. Yeah.
Okay. Yeah, maybe. Yeah, I think I was trying to replicate your walkthrough of each of the pots, but I think the important factor is just that there are relationships between natural breathing as well as deep breathing and breath holds. But importantly, those are both related to vascular density.
Yeah.
Okay. Yeah, can do. Does anyone else have questions or comments?
Would it make sense to explain those red lines on slide number 3? I was a bit bothered that I don't understand on slide number 3. So where you show the
this
breathing
3. Okay.
curve to like add some kind of small arrow with a text explaining what's happening there. Is it like breathe in, breathe out, or?
Sure. Yeah. Yeah. Just for understanding right now, these are the locations of the respiration variation, and these are natural variation. So they're not instructed changes to the breathing. They're just that like people sometimes change their breathing, fall asleep, get bored, whatever. But yeah, I'll be sure to make that clear.
Tyler, so this respiration variation is the one that, Peter Bandettini also used in the past about the respiration volume per time measure?
It's related, but it's not exactly the same. So this is specifically, Yihui did a lot of work on trying to understand vascular effects using a sequence called vapor, which tries to cancel out as much of the moving molecules as you possibly can. I haven't even really approached that aspect of it, but it's in the more extended slides, like how is this actually addressed? But did you have a question regarding that, or?
I am asking, like, when you say stable respiration versus respiratory variation, what is it like? Can you show a formula? Like, is it the inter-respiratory interval mean of that, or a coefficient of variation of the breathing, like peak distances, or is it the depth of breathing divided by the time between two minima?
I think this could relate to any kind respiration type change. For example, the depth or the period or the cycle or kind of like the phase, any kind of those respiration change.
But they were identified by hand, Yihui, or?
Yeah, this like this respiration change is detected automatically. So use some AFNI program. Yeah.
What is it doing? Just the problem is like AFNI is doing something, but as an audience, I don't know, because they are all different things, and they may, like, for instance, respiratory variations can be induced by stimulants too. And have you measured these people using any drugs or opioids or alcohol or things like that? If you don't have any controls, it will be a problem. And also, like, what type of measures, and whether or not and actually, let me jump to my second question. So those voxels that are impacted by respiration, you take it as noise in the fMRI?
We usually think it's like a kind of contributor to fMRI noise, but here we think this, for example, this respiration change, this effect on fMRI image is actually related to the vasculature.
Yes. So it has something to do, like it's not like a noise or something that it's the kind of, it has a relevance for vasculature.
Yeah, it's
Yeah,
relevant
and
to
that's
vasculature.
kind of the purpose of what he's
Okay.
talking about in this, is that the vasculature carries a lot of information about breathing and other physiological processes.
I'm not sure if you, for example, there's some tech, like some method called CVR, the cerebral reactivity measurement CVR, like look, it's actually very similar. Like for example, CVR, people can do resting state CVR. Basically, they consider during rest, people still, for example, have some respiration variations, so they can use these to induce some app called CVR. It's actually very similar, like.
CVR, you said, like the some
CVR.
volume ratio, like cardio, or like blood, is it perfusion or what?
It's called cerebral volume reactivity. Like there's a resting state CVR. Let's see if I can find it quickly. For example, like let me share screen. For example, like people also do those kind of cerebral vascular reactivity mapping, and we can do it in resting state. The assumption is during resting state, there are some respiration changes, so we can use this respiration change induced like a variation to which will reflect this CVR, and the ideal, like in our work, is actually very similar to the resting state CVR. Regarding reflecting the vasculature, like relating respiration to the vasculature.
Okay, thank you.
Yeah.